All right, everyone. Let's get started. All right. So somebody tells me I got 4 minutes for a speaker. All right. Who's finishing their dissertation. All right. All right. So let me do this quick. All right. It's a pleasure today to introduce Erin Larson, who's going to tell us about her dissertation research. Erin is from New England. She grew up in Connecticut. And in high school, she learned to dive, all right, and ever since, she's become fascinated with aquatic ecosystems. All right, she went off to college at Dartmouth, and there are a couple things in particular that stood out to me that she's talked about. One was after her freshman year, she went to Ecuador, and she taught English and math, all right, in a small village up in the Andes. Right, so that was super awesome. All right, and that was where she really started to have this passion about teaching. I'm going to return to that in a moment. All right, and then she also another year, after junior year, she went west to Rocky Mountain Biological Lab, where she did her honors thesis research on nutrient limitation of a nuisance diatom. All right, so after college, she went back west. She's kind of like a born-again Westerner. And so she's spent a couple of years working for the Idaho Department of Fish and Game as a fisheries research tech before starting grad school here in 2013. And Erin has just been this super awesome graduate student, right? And I'm really going to miss her a lot. I think everyone in her lab is. She has this unique special aura to her. But not only that, all right, it comes in this rainbow of colors, all right? All right, so however you do that, Erin, just keep doing that, all right? All right, so one of the things that Erin, is very, just upfront about is the importance of work-life balance, right? And this is something that I pirated from her website. I love this photo, all right? It's one that shows Erin at one of her field sites that we'll hear about today, all right, up in the Andes. And you can just see how focused she is. And Erin is really, really focused, all right? In terms of her research, she's probably the single student who sent me the most PDFs, all right, in the last, just saying, Alex, you ought to check out this paper. And she's actually read them, all right? All right, so that's kind of the work side to her, all right? But Erin has all kinds of ways that she has figured out how to play hard, all right? A lot of them are have something to do with the outdoors. And she's like the only student I've ever had, all right, who is a dedicated skeet shooter, all right? I'm really sorry I never actually saw this in action, all right? Okay, and with Erin, you always know how to get a good laugh with her. All right, thanks for all those laughs. All right, and then the last thing that I want to talk about is, again, Erin is this passionate teacher. And so, even though she's had really great support, NSF support, all right, Erin has carved out a lot of time, all right, as a graduate student here in terms of engaging in, active learning, these kinds of things. The last time we taught stream ecology, she was really a co-instructor for the class. And she already has a faculty position, so she's going to head off to Alaska Pacific University in Anchorage, where she's going to start a faculty position in July. So that's super. All right, and then the very last thing that I want to say is, I just have utter respect and admiration for Erin and her dedication to the Cornell Prison Education Program. So she's taught 2 classes with Michelle Wong. So thanks really to both of you, all right, for this, all right. And so this program, all right, for incarcerated students, all right, if you will, all right, people that are incarcerated And they've taught up in Auburn at the correctional facility up there. And this is just a sector of society that's often really neglected. And Erin's been really dedicated to that. So thank you for that too. All right, so with that, we're going to hear about structural and functional consequences of disturbance on biodiversity in tropical and temperate montane streams.
Thank you very much, Alex, for that kind intro. All right. Well, happy Earth Day, everyone, and thanks for coming out. I'm very excited to talk to you about some of my dissertation research. And so to start with, I wanted to show this slide depicting the incredible diversity of disturbances that we can find in ecological systems. And so disturbance plays a really important role in all types of ecosystems. Whether you work in terrestrial, marine, or freshwater, it's a really strong structuring force in community ecology. And I want to make sure that we all get on the same page about what a disturbance actually is. So there's kind of these two pretty famous definitions of disturbance. One is by Pickett and White that kind of thinks about disturbance having consequences at an individual, a population, community, and ecosystem level, affecting resources and also causing mortality. And then a sort of more narrow definition by Wayne Sousa that talks about the effects that disturbance can have on individuals and populations. I just want to make sure we're all comfortable with what a disturbance is. It's sort of a term that gets thrown around a lot in ecology before we begin. And because disturbance can have effects across the ecological hierarchy of organization, it can be really complicated to study. So this is an example of a path diagram thinking about the effects that wildfire can have on stream systems, stream food webs. And as you can see, there are a lot of boxes, there are a lot of arrows, there's kind of a lot going on. I don't need you to digest all of this, but we have this sort of main structuring force that has a lot of direct and indirect effects on multiple levels of ecological organization. And so how do we simplify disturbance? How do we understand it at a more simple level? So one way that's pretty famous that's been proposed is to think about diversity as an emergent property on the disturbance effect. So for those of you who aren't familiar with this, this is the intermediate disturbance hypothesis. So we have species richness on our y-axis and disturbance frequency, intensity, or extent on the x. And we expect the highest levels of diversity where disturbance is intermediate. So where we're not so harsh that no one can survive, but we're not so benign that we see effects of competitive exclusion. And so I study disturbance in stream systems, and I want to show you a video that I think really well encapsulates why streams are such a great, sorry, my computer is being very buggy, a great system for studying disturbance. So this is a video I took in Ecuador of one of the streams I study. This is the Quijos River, which is the major drainage. And when I look at this video, I don't particularly want to live in that stream or jump in there. But this is a very typical flood event for this system. This is during the rainy season. This is the type of flooding that occurs all the time. And so not only do we have the force of water moving downstream, but we also have that chocolate milk, those sediments that are moving in the water column. We have bed loads, we have rocks moving around the bottom. So being an organism that's living in this system, you have to be ready to deal with a lot of abiotic harshness. Another example that I want to show is that stream that Alex talked about in my introduction. So this is what this stream site Guango looked like in 2015. And then here's what Guango looked like a year later, in January 2016. This photo is taken from the same vantage point. So we have floods as a potential major disturbance in streams. We also have geomorphic events such as landslides that can shape stream systems. But despite all of this, we actually see that stream invertebrates are really diverse. So these are some examples of stream insects from Ecuador. These photos were taken by Martin, who worked with me in the field and is now a photographer in the Galapagos. I think he made some good life decisions there. He's an excellent photographer. And so these are just a very small subsample of the types of insects that we see. So there's a lot of diversity both in form and in function and in terms of evolutionary history, which I think is really fascinating, that there's all of this diversity that we can see in these really harsh systems. And so one thing I want to make sure we're on the same page about too is the difference between a disturbance event versus a disturbance regime. And so These are hydrographs here. We have stream discharge on the y-axis. Then we have day, or time of year, and then year. And you can see here, this was one particular flood event, that spike there on the hydrograph. And this system normally sees these snowmelt floods every spring. And this flood was unusual and that it occurred in the fall. So that's one particular disturbance event. Now, this idea of there being annual snowmelt flooding that happens every year in the spring is a disturbance regime. So these sort of patterns of disturbance that we see on annual and interannual cycles. Just to add another wrinkle to studying disturbance can have effects across spatial hierarchies. So you can think about a disturbance affecting a small patch of the stream bed, a whole stream reach, or an entire watershed. And so making sure that we're really explicit about the scale at which we're studying disturbance is also really important. Which brings me to my major question that I'm going to talk to you about today, which is how can we use multiple approaches to understand the effects of disturbance events and disturbance regimes on ecological communities across scales. And so I'm going to set out sort of this roadmap for us. We're going to first talk about extreme disturbance events and think about consumer resource relationships and how they change as a result of an extreme disturbance, and then diversity changes as a result of a disturbance event. I'll then move on to talk more about disturbance regimes and talk about them at small temporal and spatial scales, and then at larger temporal and spatial scales. And the systems I'll be talking about for both of these are for each of these, are in the Colorado Rockies and the tropical Ecuadorian Andes. And so in the Colorado Rockies, I looked at the effects of an extreme flood event. And then in the Andes, I looked at the effects of disturbance regimes, both in terms of landslides and local habitat stability. And as you can see from these photos, as we move along the elevation gradient in both of these places, streams look a lot different with different elevation. There's differences in canopy cover, there's differences in temperature and dissolved oxygen, which you can't see, but you might intuitively know if you've ever hiked up a mountain. And there's also differences in disturbance, and that'll be what I'm focusing on today. And so just to bring it back to our conceptual framework, I'll be thinking about and talking about the extreme flood event that occurred in Colorado, and then disturbance regimes in Ecuador and their influence. So first, let's start out with Colorado. And so in 2013, in September, there was a really major flood event that happened in Colorado's Front Range. It was a very unusual event in terms of its timing and its magnitude, which I'll get into. It was very extreme in terms of its effect on human life and property. And it was also really extreme in terms of its ecological impacts, which I'll talk about. So if we again look at a hydrograph here where we have discharge on the y-axis and time of year on the x-axis, all of the gray lines here are all other years besides 2013, and that black line is 2013. So you can see that it really stands out in terms of when it occurred, so the fact that it occurred here in the fall, and also how big of a flood it was, so how high that peak ended up being. So first, let's talk about consumer resource relationships. So there's kind of two potential ways that an extreme event could affect what food stream insects are eating. One is top-down and one is bottom-up controls. It doesn't have to be an either or, it could be sort of somewhere along that spectrum. And so for this project, I sampled 7 streams along a disturbance intensity gradient for bulk stable isotopes of both the insects and their basal resources, the food that they eat. And then I used a Bayesian mixing model to see where they were getting their food from. I also measured quantitative community composition and analyzed the results using a partial least squares regression. And so for those of you who don't think a lot about stream food webs, there's 2 potential routes through which stream insects can get their energy. One is through algae that grows in the stream or is a toxinous. And the other is through terrestrial litter or leaf fall, which is a lochthinous material. And some stream insects are thought to specialize more on algae, others more on leaves. And so changes in those relationships, changes in the amount of those basal resources could influence but what they're eating and their ability to survive. And so as a result of this event, we actually saw an influx of detritus into the stream, which was kind of surprising given the time of year when this occurred. But when we thought about it, it actually sort of made sense because it happened right before leaf fall typically happens. So we think that it introduced a lot of detritus from the surrounding floodplain into the stream rather than flushing leaves that had already fallen downstream. And I'll also show you the data, not just a photo of a bunch of leaves. So we see as we move along, the disturbance gradient here measured as a dimensionless shear stress. We see an increase in detrital quantity along the disturbance gradient and a decline in the quantity of algae. We also see that algae, there's a slight decline in their carbon to nitrogen ratio, which somewhat counterintuitively means that they're actually higher quality since they have higher nitrogen out here where there's higher disturbance, and that the terrestrial detritus is actually in lower quality out here where there's high disturbance intensity. So surprisingly though, we actually saw that insects assimilated more terrestrial resources at streams with higher disturbance intensity. So they're eating the food that's in higher abundance, but is lower quality. And that was true across these 5 different trophic guilds. And I'm not showing lines here for partially squares regression fits, but I'm happy to show those results later if you're interested. So the percent of algae assimilated in their body tissue declined with increasing disturbance intensity. We also saw that translated or corresponded also with a decline in herbivores at streams with higher disturbance intensity. There was no other significant decline in any of those other 4 trophic guilds. So overall, in the context of this particular extreme event, we see that both top-down and bottom-up controls played a role in consumer resource relationships following this event. So we both saw a loss of herbivores and an increase in the availability of terrestrial detritus. And so moving on, in our roadmap, I'm going to talk a little bit about diversity effects as a result of this extreme event. And so for this, I'm going to first walk you through a conceptual predictive framework for the responses of functional taxonomic diversity to disturbance events along disturbance gradients. But for this, we sampled 14 streams along a disturbance intensity gradient for quantitative community composition. And we did that sampling prior to the flood, 1 year after the flood, and then 2 years following the flood, and measured both functional and taxonomic diversity and analyzed our results using mixed effects modeling. All right, so I'm going to walk you through a few hypotheses. One is an idea of a low sensitivity hypothesis. So in these sort of schematics, each insect can either represent a functional combination or an actual sort of taxonomic or species identity. And the size of the insect indicates the abundance. So in this low sensitivity hypothesis, we would expect that traits and taxa are the same both before and after the flood in the community, both in terms of their identity and their abundance. The equal impact hypothesis suggests that all trait combinations or taxa experience the same mortality across the board, so they all shrink in terms of their population size in this case. In contrast with the differential impact hypothesis, where some trait combinations or taxa do better following the disturbance than others. But it's important to note that so far we haven't lost or gained any species or functional combinations, which we will in these next 2. The next is this idea of convergence or divergence. So we either lose or gain trait combinations or taxa. And then finally, we have the turnover hypothesis. So we have some sort of shift in functional or taxonomic space following the disturbance. And these convergence and divergence and turnover hypotheses aren't mutually exclusive. So you can both gain and lose taxa and experience a shift in functional or taxonomic space. And so these were hypotheses that were laid out by Kate Boersma in the psychology paper in 2016 that were originally just focusing on functional diversity alone. And so I've, as per this work, been expanding that to consider taxonomic diversity and to also put these hypotheses into sort of a predictive framework for when we actually expect to see these hypotheses occurring. So I'm going to show you that next. And so here we have a 2D space where we have disturbance intensity going from low to high on the X, and then a range of community sensitivity to disturbance from low to high. So we expect to see low sensitivity down here in this left corner, where disturbance intensity is relatively low, and there's not a wide range of community sensitivity to disturbance. I'm not going to go through all of these, but I'm going to draw your attention to the top right corner, where we see the convergence and divergence hypotheses and the turnover hypothesis. And that's what we expected to see support for in this particular system in Colorado following this extreme flood event. And so then to tie these hypotheses into actual predictions of what our data will look like, we'd expect to see either an increase or decline in both richness and evenness if we have either the divergence or convergence hypotheses along our disturbance intensity gradient in terms of the change from before to after in our communities. If we have support for the turnover hypothesis, we'd expect to see an increase in turnover along the disturbance intensity gradient. So communities would be more different from each other following the disturbance on the higher end of that spectrum. And so just to sort of see that visually for folks that don't spend a lot of time thinking about richness, evenness, and turnover, this is sort of an example of a community where we'd see evidence both for the convergence hypothesis and for the turnover hypothesis. So if this is our pre-flood community up here, our richness starting out is 3. Our evenness is pretty high. The 1 taxa represented from each species. Then if we compare post-flood, we only have 2 different species or trait combinations. And our evenness is relatively low. We have 2 representatives of that leftmost species. And if we look at comparing these 2 communities pre and post, our turnover is pretty high. These communities look very different. I also want to talk quickly about how we established a disturbance intensity gradient for this study. So we had photos from before on the left, and then after the disturbance at each site, we had 48 photos at 5 different points in the stream, looking upstream, downstream, and then to the left and right. And they were scored for evidence of channel erosion and then normalized. So this top example is one that's a very high intensity disturbance site. This bottom one, where the stream looks basically identical before and after, is an example of a low intensity site. So what did we find? In terms of taxonomic diversity, we saw a decline in taxonomic richness, also declines in taxonomic evenness along the disturbance gradient. And that persisted both 1 year after and then 2 years after the flood. So these black circles are when we compare pre-flood to 1 year following the flood. The open circles are 2 years later, or 2 years after the flood compared to before the flood. So we see this decline that persists for 2 years following. So bringing that back to our hypotheses, we see support for this convergence hypothesis. So we see these declines in richness and evenness. And now we're going to move on to look at functional diversity. I'm going to bring up a figure, and it's going to look like a lot, and I'm going to walk you through it. So this is in terms of functional diversity. We did find evidence for functional turnover and convergence, but I want to kind of pull apart our results here. So I divided our functional traits. Well, there's a previous paper that divides functional traits of stream insects into 4 categories. Those dealing with ecology, like how they eat, and the type of habitat you find them in. Life history, which deals with things like generation time. Mobility, which deals with things like how well they move around. And then morphology, so things like, are they flattened? Do they have armor? And so I looked at those kind of categories separately and then combined them all as well in analysis. So we see a decline in richness and evenness when we consider all of the traits combined, when we look at just life history traits. And then we see turnover in all of our trait combinations along the disturbance intensity gradient. And so to bring that back to our predictions and our hypotheses, we do see evidence of convergence when we combine all the traits and when we look at life history traits. Then we see evidence of functional turnover when we look at when we look at all of the trait combinations. So to bring that back into our predictive space, we do see evidence that we're in that kind of upper right corner there, where disturbance intensity is high, and there's a range of community sensitivity to disturbance. And we see a slight difference in how taxonomic diversity compared to functional diversity are responding as a result of this disturbance, which is really interesting and sort of points to why it's important to look at both taxonomic and functional diversity and think about diversity in multiple ways. So bringing it back to our roadmap, here we've found slight differences in how taxonomic compared to functional diversity are responding as a result of this extreme event. And now I'm going to move on to talk about disturbance regimes, first at small temporal and spatial scales. And so for this, I did a colonization experiment and also looked at quantitative community composition and also quantitative dispersal composition. I'll talk about dispersal in stream systems before we get going. And this was at 5 streams along a habitat stability gradient in Ecuador. This was done over 2 experimental years, and I used multiple biodiversity metrics to look at the responses at this small spatial scale, and then use generalized additive modeling to look at responses over colonization time. So I want to walk you through a couple of biodiversity metrics before we get started. And one is, so with thinking about beta diversity, you can partition beta diversity into 2 processes that are going on. One is differences in richness, and the other is differences in the identity of species who are present, or species turnover. So in this example, we have community A, we have community B, and community B is a complete nested subset of community A. So the differences between community A and community B are driven entirely by richness and not by turnover. In contrast, when we compare community C and community D, they have the same number of species present, but none of those species overlap at all. So these are communities that are entirely different in terms, and that difference is driven by turnover rather than differences in richness. Obviously, these are cartoons. Most communities exist somewhere on the spectrum where the differences between them are driven by some combination of richness and turnover. But understanding what's really driving those community differences can help us understand the processes that are distinguishing those communities. So in Ecuador, we see that local habitat stability is highest at high elevations. And the way that I measured habitat stability there is something called the Fawn-Quick Index, which was developed by a guy who worked for the US Forest Service in Montana. And the benefit of it is that it's a very easy habitat stability metric to go out and do, and it's actually been ground truthed against more time and labor-intensive and expensive methods for looking at disturbance. So basically what you do is you go to a stream multiple times, and you look at and score various categories, looking at the upper bank, the lower bank, and the stream bed, and add them all up. And high scores indicate low habitat stability, and low scores indicate high habitat stability, which still always gets me a little confused to this day. So I know looking at this, it might be a little counterintuitive, but these lower elevation streams are less stable, and these high elevation streams are more stable. Okay, I want to tell you a little bit about how stream insects move around streams, for those of you who don't think about what stream insects are up to on a daily basis. And so stream insects can move around and disperse in multiple ways. I'm not going to get into how adult insects disperse because that wasn't something that I looked at in this study, but that can also be a very important vector of dispersal as well. But a lot of times stream insects will disperse via the drift. So they basically let go of whatever substrate they're sitting on and let the water carry them downstream, which is a pretty efficient way to move around. They can also crawl around the stream bottom as well. And so what I did for this particular study was I looked at patch scale community assembly. So I sampled the background community composition and also sampled insects that were in the drift using these drift nets. And then did a colonization experiment over 23 days where I cleaned rocks, measured and labeled them, put them back in the stream, and then collected them at day 1, day 3, day 7, day 14, and day 23. And so looking at then the difference between these 3 different compositions, so looking at drift composition compared to the background community, compared to the experiment, we were predicting that our experimental composition at less stable sites would more strongly represent or resemble these drift assemblages and quickly represent the background community as well, because in less stable sites, it would benefit you to be able to move around a lot and exploit new territory quickly. And so what did we see? First, I want to talk about resource accumulation over experimental time. So this is showing something called the autotrophic index on the y-axis, which is basically the ratio of detrital resources to algal resources. And it's just an index of sort of the proportional availability of these different resources. And over all 5 streams along the habitat stability gradient, we saw a consistent decline towards a dominance by algal, primary producers over this experimental time. So in terms of proportional availability, basal resources were behaving pretty similarly. When we look at the differences between the communities that we saw over experimental time, so this is comparing that background community to the experimental composition. So each point is a pooled set of 3 rocks. Their composition compared to the background benthic composition, community composition at that site. And then colonization time here is shown on the x-axis. And on the y-axis, we have the different types of beta diversity. So on the top row, we have total beta diversity, we have turnover in the middle, and then richness differences on the bottom row. And I looked at beta diversity in 3 different ways in terms of the functional, taxonomic, and phylogenetic differences in community composition. And they all behaved fairly similarly across the habitat stability gradient. So differences between the background community and the experimental community were mostly driven by rich differences, which kind of makes sense that what you're getting on this small rock is a nested subset of what you have in terms of the potential colonizing pool. And that was consistent across functional, phylogenetic, and taxonomic composition. But what was interesting, or a little unexpected, I guess, was when we looked at the drift composition and how that differed compared to the benthic composition. It was basically the same thing. So we see that same thing where richness differences are what are predominating in terms of the difference between who we see in the drift and then who we're finding in our colonizing community, which indicates that we're really seeing the drift composition being what's driving who's getting to these rocks. So things aren't crawling in from elsewhere. It's really just that who we're finding in our colonization experiment is a nested subset of who we find in the drift. So bringing that back to sort of a cartoon look at it, both our drift and background community are sort of this wider group of species that our colonization experiment ends up being a smaller subset of. And we don't really see these major differences in the response at this patch scale along our habitat stability gradient. And when we look at community composition in a slightly different way, so this is a non-metric multidimensional scaling plot. So in this plot, points that are closer together are more similar in terms of their multivariate community composition. And each line here is connecting a group of sites. So we see that the squares, which are our colonization communities at the end of the experiment, are pretty far and clustered together, and then are pretty far away from the the background community and drift assemblages. So we do see that also these colonization communities aren't really reaching the same composition as the background communities over experimental time, and that difference isn't really driven by habitat stability. So what is driving it then? So another biodiversity metric that we can use to look at this is taxonomic distinctness. So that is the average path length between 2 species in a community along a Linnaean tree. And what we see is that high levels of taxonomic distinctness are reached early in experimental time and actually are as high as background levels. And so there's potentially a role for limiting similarity and biotic interactions being important at this really small scale, which probably is not very surprising. So overall, in terms of this experiment, we see that habitat stability isn't really a strong predictor of what's happening at these really small patch size scales. So now we're going to move on to look at what's going on at larger temporal and spatial scales and to see if disturbance could be playing and disturbance regimes could be playing an important role there. And so for this part of my dissertation, I did landslide susceptibility modeling across the drainage where we worked in Ecuador. I collected quantitative community composition across 5 years at 10 streams along the habitat stability gradient, and then used, again, multiple biodiversity metrics, multiple regression on matrices, and mixed effects modeling to analyze the results. And what I thought I would find would be that we'd see higher temporal community stability at more stable streams, and that differences in habitat stability would drive community differences along the elevation gradient between streams. And so to remind you why I might be looking at landslides, this is what one of our streams looked like in 2015, and this is what it looked like in 2016, which really hinted at the role of, or more than hinted, at the role of landslides in driving really important processes across this landscape. And once we saw this, we started to look around and realize that there were landscape scars sort of all over the drainage that we worked in. And so what I did, which I'll walk you through this brightly colored map, is used remote sensing data and publicly available data to create a landslide susceptibility map for the Quijos River drainage. And so you can think about, there's 4 sort of major drivers of landslide susceptibility. One is precipitation, slope, geology aspect can sometimes be really important as well. And all of those sort of sum together lead to landslide susceptibility. And so for this, I used a bivariate statistical approach where you build a landslide inventory based on Landsat data and ground truth data that we've had from being at the sites, and then look at the proportion. For example, if we look at this yellow band here of precipitation, look at the proportion of that area that is taken up by landslides compared to its total area, and then divide that over the total area of this map the total area of this map divided by, or with the area of the landslides on top of it. So you're basically looking at the density of landslides in this yellow band compared to the density of landslides on the whole map to come up with a weighting factor for that particular precipitation category. You then sort of sum up all of these weighting categories together to get an overall landslide susceptibility. And here on these maps, these green dots are the stream sites and the black outlines are the watersheds where we were working. And so I looked at landslide susceptibility both as a point value where our streams actually are, and then at a whole watershed scale to see which was a better predictor of community composition. And so to look at just quickly too along the elevation gradient, we do see that these mid-elevation streams are more susceptible to landslides. There's obviously a lot of noise there. So that mostly that's being driven by both precipitation susceptibility, geological susceptibility, and then that slope effect as well. And when we then actually looked at changes in community composition over time, we do see very high temporal variability in multivariate community structure. But it actually was best explained by local habitat stability rather than any of our landslide susceptibility metrics. So again, here we have this non-metric multidimensional scaling plot where streams that are closer together are more similar to each other. And if we just, and again, streams are connected by lines. So for example, this is a very low stability site here in orange. If we trace it from 2012 to 2014, 2015, 2016, and then 2017, you can see that it's moving around a lot in this multidimensional space. But if we compare that to this blue stream here, that's a high stability site, we see that it is also moving around a lot and changing a lot in terms of its multivariate community composition over time. When we look at biodiversity in another way, so here, just similarly to in the colonization experiment, we have total beta diversity on the top row, turnover, and then richness on the bottom row, broken out into functional, phylogenetic, and taxonomic diversity. We see that there's really high interannual community turnover, and that's mostly in just the phylogenetic and taxonomic composition. And so each point here is a community at one site compared to the community at a different time at the same site. And time difference in years is here on the x-axis. Surprisingly, functional composition is actually pretty consistent. So we don't see that changing much over time compared to phylogenetic and taxonomic diversity. When we then look at differences among streams along the elevation gradient, this is a very busy plot, I know. But I want to draw your attention to the fact that in the top row and in this middle row, these lines are all increasing. So the x-axis here is elevation difference. And this is pairwise comparisons of sites along the elevation gradient, but at the same year. So we see that things are different along the elevation gradient, so communities are different along the elevation gradient. But surprisingly, in our mixed effects modeling, that was actually driven by elevation difference, but not by habitat stability differences. So we do see that there's high turnover consistently across years along the elevation gradient, which is actually sort of relieving to see, because often we have a lot of studies that go out and sample along gradients, and you're never sure when you do these sort of space for time substitutions, if you went back the next year, would you see the same pattern? And this suggests that you actually might. And so one thing that I was surprised that we didn't see an effect of this landslide susceptibility metric on our community composition. One possibility when we look at this map is that our regional species pool in this drainage is actually being filtered by this frequent severe disturbance that is landslides regularly enough. So most of the drainage actually is fairly susceptible to landslides, and very sort of observationally when we were out, you could see a lot of different landslide scars around the landscape. And so it might be that stream insects in this drainage are sort of already being filtered to be able to respond to having low habitat stability. So to summarize this part of my work, we see that on large temporal scales, we see taxonomic and phylogenetic responses over time, but not functional responses. And that time is really our best predictor. So habitat stability was not a strong predictor in our models. When we look along spatial gradients, we see turnover of all diversity metrics. That's really predicted by differences in elevation between streams. And so to bring this back to the bigger framework and to remind you of what we've found so far, we saw that in response to an extreme flood event, there was both top-down and bottom-up controls were important in determining consumer resource relationships along a disturbance gradient. We saw that taxonomic and functional diversity experienced convergence, and functional diversity experienced turnover following an extreme disturbance event. At small temporal and spatial scales, we didn't actually see that habitat stability was a very strong predictor. And then at these large temporal and spatial scales, we saw that both time and elevation differences could play a role. So overall, I think one thing that my work has really taught me and has been really interesting to think about is that multiple approaches can really help us understand disturbance. So I like to do a lot of different things, and it's been really fun to be able to put together a bunch of sort of disparate feeling projects to study disturbance from these different angles. And the reason why this is important to consider is that we're currently living during a time when there is an increasing frequency of extreme events. So this is a map from a recent paper looking at the future return period in years of what is now considered a 100-year event, so precipitation event. So in some places, we're seeing these high precipitation events occur more frequently now, and in other places, we're moving towards more drought. We as humans are also altering a lot of disturbance regimes, and we're seeing disturbance regimes being altered globally. And so this is an example of another hydrograph. This is from the Flaming Gorge, when the Flaming Gorge Dam was put in on the Green River. And so we have discharge here on the y-axis, time of year, and then the year. And you can see that before the dam was put in, there would be very high stream flows during the spring into summer, and then low flows during the winter. And now things are much more evened out throughout the year. And using these multiple measures of biodiversity might really help us with conservation efforts and with improving prediction. So this is a global mapping exercise looking at bird and mammal diversity worldwide. And I want to draw your attention here to the desert Southwest, where we see high taxonomic diversity, low functional diversity, intermediate phylogenetic diversity. But then when we combine it, we can think about and prioritize diversity as we want to measure it and prioritize it. And using these multiple approaches might help us do that as well. So I want to thank all of the excellent funders who helped me fund this research, the EEB staff and research support, especially Erica Mudrak and Kim Sparks. Alex, who's been a very energetic and supportive advisor, and has sent me a Bitmoji birthday card on my birthday over the last few years. But all kidding aside, I think Alex has been one of the most supportive advisors I could have asked for, but has also given me a lot of intellectual freedom to do what I find interesting, for better or worse. And so I really appreciate how much he's given me free reign throughout grad school to do all kinds of fun stuff. My committee members, Andrea and Leroy, who are participating remotely. Andrea was my advisor in Ecuador and Leroy in Colorado. And I love these photos of them because I think they perfectly encapsulate them. Andrea is always willing to do whatever it takes in the field. Here she's drinking this water so that we can use this water bottle to hold samples. And Leroy is just such a gentle and kind teacher who's helping us learn how to use a laser rangefinder. And they've really adopted me into their labs when I've been in Colorado and Ecuador, and it's been so awesome to learn from both of them throughout the years. Monica and Todd, who are so great and help me think about community ecology and ecohydrology from perspectives not of folks studying stream insects, and encourage me to make sure that my results make sense to a broader audience. You guys have been awesome, and it's been really fun to work with you both. Obviously, this work takes a lot of people and time, and so in Colorado, I've been fortunate to have a lot of awesome folks helping me out in the field and in the lab, and I'm really grateful for all their help, including biking into one of our field sites when the road was closed and we had to bring everything in 5 miles. Folks have been just so wonderful. Similarly in Ecuador, Ecuador has felt like a second home during grad school, and it's been so awesome to just have a hugely supportive group of people to work with and learn from there. And this work would be really impossible without all their help. I really want to thank the Flecker and Hairston labs who've been really, really helpful and fun to work with, and always provide very critical but very constructively critical advice. It's fun to have your lab be your toughest audience, and I think it's really made me a better and more confident scientist, and I really appreciate that. I also really want to thank the sea pigs. So one of my favorite parts of grad school that I don't know if Alex knows as much about, getting really into running, especially ultra running. And the sea pigs have just been the funnest group of people to go and run and talk about various grad school things with. And thank you all for being such awesome running buddies and friends. The whole EEB grad community is so wonderful too. I especially want to thank Lena, Emily, and Michelle, who have been 3 of my closest friends during my time in grad school, and so many more friends here in this room and from afar who are tuning in. PhD takes a lot of people and a lot of support, and it's been wonderful to have that. I also want to thank my family who's here today, and who have been just so loving and supportive and have only occasionally asked me when I was going to finish my PhD. They've just been awesome and always encouraged me to get back up when things have been tough. And love you guys. Thank you. I'm also fortunate to have a bonus family. My in-laws have also been incredibly supportive and understanding of why I live in other places other than my husband most of the time. And so, they've also been just so wonderful. Also, my husband, Tom, who's currently in Alaska and thankfully brought us out to Alaska, which has been just such a fun adventure. He even helped me pick bugs in the field once, even though he told me he hated it and he didn't understand how I could do that. But he still did it with a smile. So he's been supportive throughout, and he's been very wonderful. And finally, this dissertation is dedicated to my brother, Alex. Some of you might have known that he passed away during my second year of grad school. And I think that for me was a moment when I really decided that work-life balance wasn't something that I could wait for. And so, yeah, the UB grad community was huge in helping me throughout that time. But I also want to encourage those of you who starting out in grad school or aren't starting out that life is short, so make sure it's fun. Go out and do fun things and make sure you're doing what you want to be doing. And so with that, I'd like to thank you all for listening, and I'd be happy to take any questions. Yeah, James.
So I noticed you had a in your example that we can call flies. Did you those responding differently, or were they all pretty much?
Oh, like did the mayflies, stoneflies, and caddisflies respond differently? Yeah, we do see them responding differently. In part, I think because of their functional traits are really different. So, yeah, they did respond differently.
I guess it was more
Post-disturbance. Yeah, so especially in Colorado, the Batis mayflies actually did really well, partly I think because they are multivoltine. They also had an adult stage present during the flood. They're also pretty scrap. They're a very weedy species. Yeah, so those were, they actually increased in abundance in some cases following the flood. Yeah.
So in your last section, you were showing how like functional diversity and phylogenetic diversity change with elevation.
Mm-hmm.
One of the things you pointed out is that the slopes were different, right? I think phylogenetic diversity goes more quickly.
Yeah.
Functional diversity doesn't go as quickly.
Mm-hmm.
What do you think causes those to decouple?
Phylogenetic, taxonomic, and functional.
Yeah.
Well, there's a couple of things. So a lot of it depends on what functional traits you use. So I ran into this with like looking at picking functional traits. If you pick too many functional traits, you can end up with the same numbers of functional traits as you do species. So each species is functionally unique, which depending on what traits you're interested in can be the truth, right? So I think some of the decoupling has to do with trait selection sometimes. So that's why trait selection functional diversity. But it also has to do with, so that some traits are more phylogenetically labile than others as well. And so sometimes you'll see that the taxa that you lose are unique across multiple functional axes and might be also like very unique phylogenetically, but they're just one species. So in terms of their functional or phylogenetic impact, that losing them might be very impactful for that index, but not for taxonomic diversity. Yeah, Katie.
In terms of like the importance of elevation for community differences, do you think, I'm hoping I'm interpreting the pairwise comparisons between sites correctly, but do you think that's more due to like just dispersal ability or versus a local adaptation?
Yeah, that's a great question. And Ron, I realized I meant to just talk a little bit more about. So I think, so along the elevation gradient, there's differences in temperature and dissolved oxygen. And so Dean Jacobson's done some really interesting work, and Andrea and Claudia as well, with thinking about oxygen as being something that structures communities along the gradient. And then Alicia Shaw, who did a PhD with Cameron Gallenbohr, looked at how temperature might structure differences in, so basically thermal adaptation differs along the elevation gradient as well, especially since in Ecuador there's such low temperature variation throughout the year. Those insects might be very sort of thermally adapted to exist on a narrow band of temperature. So I think the reason that elevation difference ends up being a really good predictor is that it sort of incorporates both habitat stability, since that is related to elevation, temperature, and dissolved oxygen. So it's sort of a more holistic measure of abiotic differences than habitat stability alone. Yeah.
You mentioned the kind of issues sometimes with using space for time substitutions. And one of the things that I thought was so amazing about your Colorado data set is that you had the pre-data.
Mhm.
So I have two related questions. One is like, how is it that you had the pre-data? And the related question is, I think that most of what you presented was then changed from before versus after. But my question is along that disturbance gradient, how different are those streams and their communities themselves, just irrespective of that major event? I see why you did it the way you did it.
Right.
Like, given that there's over, let's say, a 200-year period, probably several of those major disturbances on the disturbance end of the gradient, not so on the other. If you just plotted like the pre-communities against the disturbance gradient, how different were those?
Right. So I'll answer that first question first, and then the second question. The first question, I was fortunate to be part of the EvoTrack project that Alex and Kelly, and then other folks in Colorado and Ecuador have been a part of. And so the pre-data actually comes from Rachel Harrington's master's thesis, where I'm looking at community composition. In terms of the second question about how, like, if I just look pre, so when I did the mixed effects modeling, the pre-value was actually a predictor to control for the pre-effect. Because when you just look at the raw data, things are just sort of all over the place. And in terms of the question, what I was really interested in was the change. Like, do we see a shift versus just the raw value? Yeah. And another thing I just want to mention too about that flood event is it was actually a pretty, so our, in terms of the precipitation, our gradient design incorporated or encapsulated from a 1 in 50 year to 1 in 500 year event. So it is a pretty extreme event in terms of the rain event and then subsequent flooding. Yeah, Harry.
What are your main teaching obligations? Good job.
I'm still working that out, but it looks like I'll be teaching field methods in ecology, conservation bio, and interbio. Not all at the same time. One and then the other. Yeah. Yeah.
All right, let's thank Erin.
Hey, sorry. Hey. I missed you.
Sounds good, yeah.
Yes.
We'll see. I did a talk.
All right. Hey, Sam.
Thank you. I'm just gonna stop the live streaming real quick. All right. Hi, people. Bye, everyone.